Hello and welcome to pH Biology. Today's explainer is the antitoxin, a specific protein that neutralises toxins released by bacteria. By the end of this video, you'll know how antitoxins differ from antibodies, why some bacterial diseases need antitoxins specifically, and how vaccines exploit this. An antitoxin is a protein produced by white blood cells that binds to and neutralises toxins released by bacteria. Specific to one toxin, the same lock and key principle as antibodies. The difference, antibodies label or neutralize pathogens themselves. Antitoxins neutralize the poisons that bacteria release. Three things. 1. Antitoxins target toxins, chemicals, not the bacteria themselves. 2. The same lock and key specificity as antibodies. And 3. Important for diseases where bacteria release damaging toxins, even if the bacteria themselves are killed by other means. Common slip. Antitoxins aren't antibodies, even though both are proteins made by white blood cells. Antibodies bind to pathogens or their antigens. Antitoxins bind to toxins, chemicals released by bacteria. Quick example, tetanus is caused by a bacterium that releases a toxin damaging nerves. Even if the bacteria are killed, the toxin can still cause paralysis. Antitoxin neutralizes the toxin. That's why a tetanus vaccine contains the inactivated toxin, training the body to produce antitoxin in advance. Right, if any of this is going to stick beyond this video, drop into phmaths.com. You'll find a free 4-week GCSE higher revision planner ready to download, plus the mastery workbooks. One for every higher topic, every answer fully worked, every common slip flagged. Simple explanations, serious results. See you next video.